Next, I welcome Jasna K from Jamia Nadwiyya Training College to talk about assessment of tree diversity in Gandhigram Rural Institute, deemed to be University Dindigal, Tamil Nadu.
Good morning, everyone. Today, I would like to speak about the assessment of tree diversity in Gandhigram Rural Institute, Dindigal, Tamil Nadu. First of all, I'd like to speak about the biodiversity. The biodiversity is the word which is invented by the many experts in the field of biology. And biodiversity is the word which is more meaningful after Edward O. Wilson in Harvard University introduced the book entitled as Biodiversity, which is an intention of biological diversity in 1989. There are mainly two important factors which causes the loss of biodiversity. That is, one is natural and other one is anthropogenical activities. In natural activities which include earthquake, landslides, flood, etc. And in case of anthropogenical activities which include study based on the radiations and also the nuclear fission, fusion and the deforestation and urbanization. Then the word biodiversity indices. These are the mathematical expression which is used to identify the special features like the dominance, recessiveness, which species is more dominant in a selected area, which one is more recessive, which species is going to extinction, etc. Then this is the map which shows that I selected the area for my project. The area from Dindigal district, Tamil Nadu, Gandhigram University. Then major materials and method. The tree inventory method was adopted for the collection of the tree data, and species diversity, species dominance, species evenness and species richness will be calculated for the identification of which species is more dominant in an area, which one is more rich in that area, then the relative dominance of that area. By the analysis of these mathematical expression, we can able to identify the dynamics of the ecosystem. Then the result of my study. The total number of 109 tree species in each 34 families are be identified and 2512 number of individuals were be identified and recorded from the study area. Azadirachta indica has highest percentage of distribution among 739 number of individuals. Azadirachta indica, which is vernacularly known as vape, which means neem. About 90 species only have one number of individuals, which include Citrus medica, Butium, Monosperma and Ficus racemosa, etc. This is the graph which shows the number of individuals, including in each families. Here it is clearly shows that 1019 number of individuals will be present in Fabaceae and it is followed by 742 number of individuals present in Meliaceae. So the family Fabaceae have more number of individuals present in our study area. This graph shows the relation between the number of individuals and the families. The family Fabaceae, which have 28 number of individuals, there is more number of individuals which is including in the family Fabaceae. These are the mathematical expressions that are used to identify the specific features like which species is more dominant, which one is more recessive, which species have more number in that area. The Shannon-Weiner index, the calculated value is 2.61, which means the diversity in the study area is more. When the value is greater than 1, which indicate the diversity is more. Then when the value is less than 1, which indicate there is no any diversity. Here it is clearly shows that the value is 2.61, that is greater than 1, which indicate the diversity is more in the study area. Then in next one, Simpson index, which is used for the identification of the species diversity. That the value is 0.2639. It is ranges in between 0 to 1. When the value is 0, there is no diversity. When the value is 1, it is indicated as there is an infinite diversity. So it clearly shows that the value is in between 1 and 0 to 1. So it indicates there is a moderate diversity present in the study area. When In case of species evenness, which indicates the relative dominance, which species is more dominant in the study area than the area which consists of more number of species. And the species evenness have the value 0.639, which is ranges between the 0.5 to 0.8, which indicate the moderate number of diversity in the study area. Then the species richness. The species richness is 2.0460 is the calculated value, which is ranges in between the 0 and 5, and it indicates the moderate diversity present in our study area. Then finally the conclusion of my study. The diversity of tree species and their indices in the study area has been documented. It has been shown that the diverse tree species are necessary component of the institution. There are several anthropogenical activities which were found in the university, which causes the major reason for the decrease the number of species in the selected area. And the present study, which recorded 2512 number of individuals and also 106 species and 34 families, provided the heterogeneous tree composition in the study area. Tree inventory and the survey should be conducted on area to note the biodiversity indices. The present work, which is helped to provide what are the major managemental strategies for the conservation of the study area, should be provided. Thank you.